So glad to welcome everyone to the Memorial Symposium for Eric D. Young, from the auditory nerve to cochlear nucleus to inferior colliculus to auditory cortex and back. I'm glad to see many of you here today for the symposium, and I'd also like to highlight that Eric's family, his wife Pam, and their son Daya are also here together with Elizabeth. And finally, before we go on to the symposium, I'd also like to acknowledge the sponsorship from ARO, from the Johns Hopkins Department of Biomedical Engineering, and the Edmund and Lily Safra Center for Brain Sciences. So just a brief overview of what we're going to talk about today. First, we will start off with a brief intro by Xiaoxin Wang, professor and colleague, longtime colleague of Eric's at the Johns Hopkins University Department of Biomedical Engineering and Center for Hearing Sciences, and he will speak to Eric's contribution to Johns Hopkins hearing research, the ARO, and to the success of his many colleagues. And actually, I should mention that you might have seen the printed version of the schedule that Paul Fuchs was originally scheduled to present this, but he was unable to come. So Xiaoxin has stepped up. Thank you. And then Laura Carney, colleague, will speak to Eric Young's contribution to quantifying and modeling the neural representations of sound. And then Ian Bruce and Mike Kahns will talk about auditory nerve encoding with acoustic trauma. Then Ellie Narkin will be speaking about the dorsal cochlear nucleus, the functional anatomy and circuitry. And then I, Lena Reese, will talk about beyond the STRF, random spectrum stimulus models. And then Patrick Kennard will talk about multimodal process in the mostly dorsal cochlear nucleus. And then we will conclude with Alan Palmer, who will talk about the international influence of Eric and his science. And then we'll have some closing remarks from co-chair Tilak Ratnanatha about the Eric Young Memorial Fund to support students with hearing loss. So I'll now give over the podium to George, who will introduce the first, I am introducing. Okay.
Yeah, can you hear me here through this? Okay. So our first speaker is Xiaoxin Wang. Xiaoxin come up from Hopkins. And Xiaoxin was a graduate student with Murray Sachs, and whom many of you know was Eric's career-long friend and collaborator. And it was a very unique scientific relationship. Murray and his wife Merle, and Eric and Pam, raise your hand, were very close friends. And that good feeling trickled down into the laboratory. It was like a trickle down that worked. And also, by the way, Xiaoxin is receiving the Pioneer Prize at this meeting, so make sure to attend that session also. So, Xiaoxin.
Okay. So, thank you, Nina and George, for organizing this symposium. I'm Xiaoqin Wang, a professor of biomedical engineering at Johns Hopkins. I speak here today on behalf of Paul Fuchs and Eric's colleagues to give you a picture of Eric's role at Johns Hopkins. Beginning with his doctorate with Murray Sachs in 1972, Eric spent his entire career at Johns Hopkins. After a postdoc with J. Goldberg at the University of Chicago, Eric returned to Hopkins as assistant professor of biomedical engineering in 1975, rising through the ranks to become professor in 1987, with joint appointments in otolaryngology, head and neck surgery, and neuroscience. So here you see a picture showing Eric with Murray Sachs on the left and Maurice Goldstein on the right. Maurice founded the Neural Equity Lab in the Department of Biomedical Engineering on School of Medicine campus that many of you in the audience have been too. Murray Sachs becomes the head of the neural encoding lab after Morris moved to electrical and computer engineering department on the home of the campus. Later on, Erk took on the directorship after Murray becomes the head of the biomedical engineering department in 1991. So here's a picture showing younger Erk and Murray. This picture was taken in 1978. This will be 3 years after Erk became assistant professor in Johns Hopkins biomedical engineering department. Erk worked with Murray shoulder by shoulder in the neural encoding lab. Together, they did the landmark experiment showing how auditory nerve fibers encode speech sound, which you will hear in the speakers following me. Now, this is a picture of Erk showing on the Johns Hopkins magazine many years ago. Erk holding a cat, experimental animal he worked on for most of his career. I want to say a few words about the leadership of Erk in the Center for Hearing Science, in addition to his scientific contribution, which others will speak later on. A major contribution of Erk is his leadership in the Center for Hearing Science, which was founded by Murray Sachs as a collaboration between biomedical engineering and otolaryngology. After Murray becomes the director of the biomedical engineering department, Erk took over the leadership of the center and served as its director between 1991 and 2005. Under his leadership, Erk continued and grew the center to include the faculty from School of Engineering, Medicine, Arts, and Science and Public Health. The center served as an intellectual home of the hearing research across Hopkins with weekly research seminars till today. As you might imagine, it takes patience and diplomacy to promote an interdepartmental center, balance competing interests, and encouraging faculty to participate in seminars, teaching, mentoring, and community work on top of responsibilities to their home department. Importantly, Erk served as a prime mover and a program director of center funding, including a research and training center RTC grant for 10 years, a T32 for its first 10 years, and this grant will still keep it at the center entering its 40th year, and also a P30 program grant for 15 years. Many of you in the audience today were trainees of the center and benefits from these grants. There are challenges, of course. The center itself never had a direct institutional support, salary, or tenure faculty positions. But Erk had his vision to work with department leaders who committed to relevant faculty hires, lab space, and partial staff support for new hires. And Erk promoted within-center collaborative support. By his own example, Erk was the main supporter of an electronics workshop and technician available to all center labs. Erk, like Murray Sachs before him, provided spirit and vision. He did this by his willing generosity, but most importantly, by simply being present. You could always count on Erk to be there. By his example, Erk motivated faculty and students to view the center as a supporting home for their scientific academic identity. Not only was Erk absolutely reliable in attending center seminars and courses, but he was reliable present in his office or the lab. He did not become a dean or split his time with second jobs elsewhere or seek out demanding consultancies, but rather was available whenever anyone needed his advice, be they student or professor. The Center for Hearing Science grew into Center for Hearing and Balance as we know today, with new faculty and research directions thanks to the foundational years provided by Eric Young. It's fair also to say that the more recent success in gaining substantial endowed philanthropy for hearing research at Hopkins would not have happened without the established success of the center that Eric directed for over 20 years. So some of you might have seen the picture this morning in the presidential symposium. Beyond Hopkins, Eric was a lifelong member of ARO. He served as the associate editor for the journal Auditory Neuroscience. For those of you who remember it, it's a short-lived journal. And Eric also is a founding director-in-chief of ARO, as you can see from this picture, of General ARO. Now, Eric has made tremendous contribution to teaching and education, besides as an experimental scientist. He is a dedicated educator, one of the best teachers at Hopkins. He directed a core course called Structure and Function of Auditory and Vestibular System for many years, and continued to teach lectures in this course even after his retirement. This is a flagship course on hearing and balance at Hopkins, and perhaps the most comprehensive course on the subject anywhere. Every student and postdoc who work on auditory and vestibular system at Hopkins have taken this course, including many in this audience. Eric also had a major teaching role in biomedical engineering that included modeling the auditory system, models of neuron, theoretical neuroscience, as well as participation in the BME core curriculum. The course, Models of Neuron, become a legendary course in the BME program for the last 30 years. In addition, Eric served a tour of duty as director of an undergraduate program in biomedical engineering, and later on the director of a BME program, PhD program. He additionally directed a T32 training grant for BME, in addition to the hearing research, T32. Served on professorial promotion committee at Hopkins and on several chair search committees, including the one that appointed Loie Manor as the chair of otolaryngology department at Hopkins. As some of you know, Loie Manor later on became the provost at Hopkins and then the dean of Stanford Medical School. Now, in preparation for my speech, I tried to find a picture of Eric with me and others. Here's a picture I was able to find from my auditory meeting in Prague, 2003, more than 20 years ago. You can see here in this picture, Eric sitting on the back. To his left was Alan Palmer and George Spears, I believe, wearing a glass. And you can see me in the front row in my younger days. Back then, for younger students, there was no smartphones. Pictures like this had to be taken by professional photography. Sometimes we'll get a paper copy. I might have scanned this picture sometime and leave it on a computer. Now, iPhone actually come out a few years after this. And then one day I saw Eric having an iPhone hanging on his belt. I thought the time has changed. It shows Eric's test as an engineer and a new technology adapter. So when Eric retired in 2015, colleagues at Hearing Science Center donated money for him to buy an iWatch. I think he enjoys this very much. Finally, I want to see my affiliation with Eric. I've known Eric for nearly 40 years, first as a student and later as his colleague. My first encounter with Eric was in the spring of 1986, 39 years ago, When I went to Hopkins for an interview by the PhD program, I didn't have money to buy a flight ticket. So I took a Greyhound bus from Ann Arbor to Baltimore, 17 hours. On the bus, I read this classical paper by Eric and Murray on temporal coding of speech. I still teach students this paper in lectures today. Eric is a true scholar, a dedicated educator, a trustworthy leader, a warm-hearted colleague, and a role model for scientists, both young and aged. I often told colleagues and students that you can trust every word Eric says and every paper he has published. This is the highest standard for us as scientists and teachers. Last, I want to say that for many years, the annual Christmas party that Eric and his wife Pam very kindly hosted at their home, complete with high-class food, a wide range of wines, artisanal beers, and jazz combo, have left long-lasting and permanent memory in the mind of everyone in the Hopkins hearing community, including many in the audience today. Thank you, Pam. So that's my comments to Eric. And this is a picture showing Eric in his late time in the wild, probably west somewhere. I like this very much, and we'll miss you, Eric. Thank you all for listening.
Okay, thanks, Xiaoqing. Our next speaker is Laurel Carney from University of Rochester. Laurel's interests, career-long, have had quite a lot of overlap with Eric's. And although not a trainee of Eric, I can say that Eric respected her work greatly and valued her as a colleague.
Oh, thank you, George, and thank you, Lena, for organizing and inviting me. It's a real pleasure to go back and look again at some of Eric's work and talk about it to this group. So when I was entering the field, Eric was really hitting his stride as a young faculty member, and we knew his work very closely. So I was in Madison at the time, and we studied every paper as it came out and quickly implemented all of the analyses that he suggested and tried them out on our own paper, probably before they were warm. I mean, before they'd cooled down, we were implementing them. And I'm going to talk for a few minutes about two studies that were briefly mentioned by Xiaoqing that were particularly important to the field. These are studies of neural coding of vowels published in JAZA, both in 1979. And these are still required reading, not only at Hopkins, apparently, but also in our courses at the University of Rochester. The first was encoding of steady-state vowels by discharge rates. So he and Murray picked 3 vowels, pictured here. These were cardinal vowels. So these are vowels that occur in nearly all the languages of the world. These were not randomly selected English vowels. The choice of these vowels showed to me that either they know some linguists or they know some linguistics, but they're very cleverly selected for nicely sort of spanning the space of vowels that are important to understand. And the key differences from vowel to vowel are in the frequencies of the spectral peaks. So you can see that the first spectral peak, F1, varies systematically across their set of 3 vowels, and the second peak varies going in the other direction. So the big question is, how are these peak frequencies encoded by the auditory neurons in response to these 3 sounds? So in that first paper, they evaluated the most basic code, that is discharge rate of the neurons as a function of the tuning of the neurons or the characteristic frequency, CF. And this code works great at low sound levels. But as the sound level increased, as they showed, the code breaks down. So it's no longer possible to, not sure how this works yet, to you can visualize where the peaks are based on this rate profile, but that breaks down by the time you get to high sound levels. So here the profiles are all superimposed. And as they pointed out, you can't completely count out rate coding. By working very hard, averaging together lots of neural responses and so forth, you can eke some information out of it. But qualitatively, this code is worsening over the range of sound levels where we engage in speech communication, listening to music, and so forth. And over that same range of sound levels, performance of listeners in discriminating these vowels improves. So there's sort of a mismatch in terms of how well the code works and how well listeners work. So let's see. This is just summarizing the results for all three vowels. I like looking at this slide and just imagining the number of hours that were involved in compiling all of the data that went into these plots. But in general, you'd be hard-pressed, I would argue, to discriminate these three vowels, one from another, at the higher sound levels, and at the levels that we choose to use for speech communication. And as they pointed out in the paper, when the sound level fluctuates, as it does in real speech, in real environments, these codes based on the discharge rates of neurons become quite unreliable. So those conclusions motivated moving on to the next study, Young and Sachs, 1979, which focused on the temporal discharge patterns in response to these same three sounds. So this is the same data set. This was published just a few months later. It's actually pretty remarkable when you look at the fine print. It was submitted in mid-June and accepted for publication in mid to late July. This was back when you used snail mail. So most of this time was taken up by the post office. Okay, so they studied timing. I'm not sure, and I'm kind of curious to ask if anyone knows whether Eric was more invested in the temporal study or if it was just a division of responsibilities between he and Murray across the two papers. But in general, at that time, auditory nerve coding had been studied for many years, but mainly for responses to pure tones. And the codes that had been studied were, as we discussed for the first paper, average rates, so the average number of spikes over the whole time course of the stimulus, or the synchronization coefficient, which puts a number on how tightly the spikes are synchronized to the stimulus. And that was a metric that Eric, no doubt, became familiar with during his postdoc in Jay Goldberg's lab. He actually had used it to study vestibular neurons' responses to low-frequency sounds. But it's a lot more complicated to apply this to speech sounds. So these sounds consist of many different components as opposed to a single tone. So in particular, all the harmonics of a single fundamental or all the multiples of that frequency are included. So how to pin down the temporal information in these responses, that was the first challenge. And at moderate to high sound levels, single fibers would be driven by many harmonics, which beat with each other and so forth. So they devised strategies to do just that. Right? So the first few figures in the study show that at low sound levels, the code is relatively straightforward. Neurons are effectively stimulated and synchronized to the harmonic that's close to CF. So they showed here the synchronization coefficient for responses, and they looked at it one harmonic at a time, so that they show several examples here. And in particular, you can see that phase locking of the neurons, again at these low sound levels, is dominated by one harmonic near CF, and I think it's cleanest shown in the right-hand column. So you can see the synchrony is high right when the neuron is tuned near that harmonic. So that's great. But when you go to higher sound levels, of course, it gets more complicated, right? So as the level increases, the neurons were often dominated, the responses were dominated by harmonics that could be further away from their characteristic frequency. So in particular, the synchrony was captured by the highest harmonic in the general region, which was, in most cases, that of a formant. So the three circles here show the formants, the first formant, the first large spectral peak, the second one is here, and the third one is here. And you can see that at these frequencies, a large range of characteristic frequencies are all synchronized here, here, and here. We're synchronized to the formant nearest the spectral peak. So this concept of capture has turned out to be, it's quite nonlinear, and it's turned out to be quite important to many of us over the years since. They went on to develop a metric we'll look at in a moment, the average localized synchronized rate, to try to pull together these population responses. So in particular, they looked closely at the timing using period histograms. So this is one cycle of the stimulus, and they created period histograms. This is 4 different fibers shown here. Of course, one period of the stimulus contains even numbers of, or integer numbers of periods of several harmonics, or all of the harmonics. So by studying the period histogram, you get a picture of that, but by then taking the Fourier transform of the period histogram, you can look and see whether a neuron's timing is dominated by, sorry, I've lost my mouse, there we go, dominated by one or a few harmonics, or whether it's influenced by many, many harmonics, as in some of these cases. So these visualizations give you more intuition about how complicated the coding is, but in how in some cases neurons could be quite dominated by smaller numbers of harmonics, which sort of cleans up the story in a nonlinear way. The metric they derived, which was mentioned briefly earlier, was the ALSR, average localized synchronized rate, which was just an average of the synchronization coefficients for a few neurons with characteristic frequencies surrounding each harmonic. And you can see just by looking at this plot, I hope, that there are a few peaks here, and across this really wide range of sound levels, this peak persists. So this representation does not flatten out and degrade as you go up in sound level, which was shown in the earlier study of discharge rate. This is showing the results for 3 vowels, and you can see that the peaks coding F1 and F2, again, are preserved over the entire range of sound levels that they studied here. Now, comparing those results, which are on the right, just compiled slightly differently, to the rate coding results on the left, again, you see how robust the ALSR is over a range of sound levels. So this was a major finding and taught us a lot about the value of the temporal information in these data. One question still remains, which is how would the nervous system decode this information? So that's something we still are chewing on to this day. One thing I particularly like was a follow-up study that was published the next year on nonlinearities in speech coding. And as a lover of nonlinearity, this paper stands out. What are shown on the right are figures showing the rate curves, which you can see are the ones that are sort of flattened out at these higher sound levels, in contrast to a temporal representation. So during that era, perhaps because most of us were trained as engineers, nonlinearities were a bad thing, generally considered to be a limitation on neural coding, something that we had to somehow work around. But in this study, backing up, this was before we knew about the changes in length of outer hair cells and the importance of that sort of nonlinear mechanism in the inner ear. But in this paper, they described how these nonlinearities, including influence of auditory nerve rate saturation and two-tone suppression, actually improved vowel coding and contributed to this very robust code that persisted over a wide range of sound levels. So we're pursuing vowel coding in a little bit of a different direction now, but it's really supported by this 1979 data set. So we've focused in on coding to the fundamental frequency. So instead of looking closely at what's happening near CF, as would be shown in all of these other plots, we've honed in on the code here. And if I zoom in on this picture, the key is that when neurons are captured by other harmonics over here, that means they're not phase locking to the fundamental frequency. So again, zooming in on this, you can see that when I'm borrowing the rate profile here to show you where the spectral peak is, but where you have a strong rate coding of the spectral peak, you have a dip in the response that's synchronized to the fundamental frequency, same for F2. So why pay attention to that instead of to the energy near CF? And that's because it's easy to decode. The central nervous system has many neurons that are very sensitive to low fluctuations, low frequency fluctuations. So this is a form that we think is easy to digest if you're the central nervous system. And therefore, this temporal profile described at the level of the auditory nerve gets mapped into a rate profile up at the level of the midbrain. Now, the story is not quite this simple, and we're still working on it, but I bring it up as an example of that this data still lives on. You know, we're still gleaning, I think, new ideas for neural coding just because of the sheer quantity of the data and the quality of the data and the careful choice of stimuli and so forth makes it just a living data set that will work for us for many years, many more years. Another aspect of Eric's work that has been critical in the field is its value in developing computational models for auditory nerve responses. And this is a couple of examples. I think you'll hear more in the next talk about the valuable data sets in ears with and without hearing loss. We also took advantage of some of Eric's thesis work on fatigue, so that he measured these very long-term recordings. It's hard to believe they could do this, but over very long recordings, over many, many seconds, minutes, to understand how adaptation of the responses persisted over long times. And that information we could directly use as we developed the newest aspects of our model, which are an improved synapse model. So again, a lot of this work is so valuable to us as we continue to improve our understanding and our ability to model these responses. So I began collaborating with Eric's group through Ian Bruce back in the 90s, and their input was really important in pushing us in our modeling work, and we continue to collaborate to this day. We have a few other connections, although I never worked at Hopkins on that group, but I sent them my firstborn graduate student, Mike Hines, who we'll hear from next. Another outstanding student also went there to do a postdoc, Paul Nelson. And currently, we're hosting a postdoc who's Eric's scientific granddaughter. And that's Swapna Agarwala, who's here at ARO. So we've had ties over the years. In addition to that, I've had the great opportunity to visit Hopkins more than once to give talks. And it was always a great experience. You knew that you would be challenged there. It wasn't not going to be an easy go, but we were never grilled. It was always a real pleasure. You'd come away from those visits with lists of ideas, things to test, things to try. And I remember Eric as being so serious, and yet he was really a pleasure just to chat with. So I think of his work on neural coding as a taking off point for our generation. But it was never the end point, and I don't think he saw it that way. He was incredibly supportive of new ideas, haywired ideas, everything. You know, he played a part in some cases, challenging or disproving them, but also supporting us to continue coming up with new ideas. And so I'm echoing exactly what Xiaoqin said. He was just an outstanding role model for all of us, and we miss him. Thank you.
Thanks, Laurel. Our next speaker is Ian Bruce from McMaster University in what is apparently soon to become the 51st state. So Ian was a postdoctoral fellow with Eric right around the turn of the century, which makes him sound older than he actually is. But
Great. Thanks for the opportunity to speak. And this is actually a shared talk with Mike Heinz, who was a postdoc after me in the lab, continuing on this work looking at auditory nerve encoding with acoustic trauma. But first, I do want to acknowledge that after this seminal work in the 70s and 80s in vowel encoding in the auditory nerve in normal hearing, the work was recommenced in the mid-90s before I came. So in the mid-90s, there was a postdoc, Roger Miller, who was the lead trainee amongst a group of graduate students who looked at the effects of acoustic trauma on the representation of the most studied of those three vowels, this vowel air, and also then looked at the discriminability of different vowels, how that's affected by acoustic trauma. And in this, they found that some of what was seen seemed to be not due just to the acoustic trauma itself, but having to present the sounds at higher levels because of the loss of sensitivity due to the hair cell impairment. And so they separately studied the effects of high sound levels in normal hearing cats. They also then started looking at hearing aid amplification schemes, so more standard schemes, and then trying to come up with novel contrast enhancement schemes to try to do better than conventional hearing aid schemes. And I'll just summarize some of the results here. So Eric was always looking for new and improved ways to visualize the data. So some of what Laurel showed, where they had multiple panels looking at the response to the different harmonics. I'm not sure who in the lab came up with this approach, but they realized that they could use this box type plot to combine it all into one. So now they're plotting fibers with different best frequencies along the x-axis, and now the different harmonics along the y-axis, and then the size of the box is indicating how strong the synchrony is. And this is actually in these normal hearing animals. This synchrony capture shows up quite well, so outlined in these green boxes, that you get this spread of synchrony to, when it keeps going, spread of synchrony to F1, the first formant, with a range of best frequencies around the first formant, and then similarly fibers tuned around F2 and around F3. With animals with noise exposure, with a high frequency hearing loss, and just flat amplification, so a higher presentation level, we get this loss of synchrony capture, so we get this spread of excitation, and also a lot more response to the other harmonics in the vowel, indicating a loss of the nonlinearities that are there, that Laurel mentioned are important for forming this normal representation. As I mentioned, they tried out looking at the effects of hearing aid amplification here, so one that was just based on the threshold shifts in the animals. And what they found was, when comparing it to flat amplification that's shown on the left, they could, through this amplification, limit the spread of the excitation to the first formant, but not limit spread of excitation to F2 and F3. And at the edge of this hearing amplification, so gain profile, it could introduce these responses here. You can see in the vowel spectrum here, this edge of the gain frequency profile introduces a little erroneous peak here that gets picked up in the synchrony. So showing the limitations of conventional approaches to amplification. So the conclusions from this physiology was that impaired vowel coding is due to broadening of cochlea and hence auditory nerve fiber frequency tuning, but also through careful work done once again looking at these cochlear nonlinearities, seeing that it's not just broadened tuning, it is also loss of these important cochlear nonlinearities. And also, as I mentioned, there's higher than normal sound pressure levels that are required to compensate for the threshold shift. So I'll just show quickly one figure from the Wong et al. Paper showing the synchrony for a fiber that's tuned around the second formant frequency. So at a conversational speech level, you get this strong synchrony to the nearby formant. But as you go up in presentation level, it starts responding to the other harmonics. And at very high presentation levels, it actually now starts responding to the first formant instead of to the second formant. So we get abnormalities just because of the high presentation levels that are required because of the hearing loss. They started looking at novel hearing aid schemes, as I mentioned, but there was an interest in being able to generalize this to running speech and to do more thorough testing. And so they saw that the hearing aid amplification needs to be shaped according to each phoneme's frequency spectrum, not just the audiogram. To do this more extensively in animals would be difficult, and so there was an interest in developing computational models that could help in understanding the mechanisms that were seen in both the normal and the hearing impaired data, and then also designing the improved amplification scheme. So I joined the lab in 1998 as a postdoc after Roger had finished up. And a few things that I learned during my time. One of the immediate things I learned was that Eric was very egalitarian, and office desk assignment was based on time in the lab. So when I arrived, there was a longtime friend of Eric's, Irv Hafter, so shown here is Eric, his wife Pam, and then Irv Hafter and his wife here on a tour at a conference. So Irv was finishing up a one-year sabbatical when I arrived, and even though he was a senior professor, a good friend of Eric's, he was the lowest rung on desks in the main lab, and that meant I started out in a desk in the hallway for a month until Irv finished his sabbatical and left. I must say, otherwise, I was made to feel very welcome, and it was a great place, but it did show that Eric was very egalitarian in many respects. He was, as was mentioned, also very multidisciplinary, encouraging depth in both the physiology and engineering. He was very supportive of trainees and promoted collaboration. So as Laurel mentioned, I started working with Laurel around this time, trying to use the VAL data to inform the normal hearing model that Laurel was working on an updated version of that in her lab. And the thing you'll note here is that neither Eric nor Murray's name appears on this paper, so even though I was funded by them, supervised them, Eric was happy for me to collaborate with Laurel and for us to get the recognition for the normal hearing model. We did then come out with, in 2003, the first version of the hearing impaired model, where we were able to incorporate both outer hair cell and inner hair cell impairment. This model didn't fully explain all of the data that Roger Miller and colleagues had collected. So when I moved to McMaster, I managed to recruit a great PhD student, Mohammed Zalani, and we were able to come up with a couple more refinements of the model. So one, an improved main nonlinear time varying filter for the model that Laurel's lab had been working on in the intervening years, and then also adding in an additional broadband filter that was explaining what was happening at these higher sound pressure levels. So So my lab has continued to work on this model, as well as Laurel's and Mohammed Salani's, and many people are now using these models. So I want to finish up there and hand over to Mike Heinz to continue the story.
Great. Yeah, so I joined Eric's lab in the fall of 2000, coming from Laurel Carney's lab, and had done computational modeling of physiological responses relating to perception, but I'd never actually done a physiological experiment. And so it was, I'm still a bit surprised Eric took a chance on me, letting me come to the lab, but seems to have worked out okay. And 20 years later, I'm still doing auditory nerve recordings and taking chances on other students who are engineers and not having done physiology before. So, but I was interested in studying hearing impairment, and the model that Eric talked about with the noise-exposed cats was attractive. And so Eric and I decided to work on sort of a small, minor issue with hearing loss, easy to explain, loudness recruitment. And so, the idea that for normal hearing, here on the left, loudness is growing steeply and then more shallow, and then with hearing loss, it grows more steeply and catches up to normal at higher levels. It has big implications for hearing aids, not being, wanting to amplify soft sounds, but not too much at high levels. But the neural correlates of loudness recruitment were still uncertain. And so if you look just on the basilar membrane, it all seems to be a pretty simple story, that same kind of responses seen on the basilar membrane as the loudness functions. But there's this pesky little thing between basilar membrane and the brain, auditory nerve. And so rate level curves that are limited in dynamic range, different spontaneous rates, different thresholds, that kind of thing. And so we wanted to see if the sort of general, sort of simple hypothesis that loudness is steeper, so rate level curves must be steeper, it doesn't quite make total sense, but we wanted to characterize this. So we recorded populations of noise-exposed auditory nerve fibers, and we found that, not surprisingly, that they weren't always steeper, but surprisingly, they actually were shallower, which, at first wasn't really what we expected. We thought they wouldn't be as steep as predicted, but they're actually shallower. So this is a population of nerve fibers, and then blue is normal, and then green is mild hearing loss, and red, more moderate hearing loss. And you see that they're shallower near the noise exposure frequency. So, we wondered why, there was something wrong with our exposure or something weird going on. And in classic Eric style, we thought about how to really get more insight out of our data. And so we used this technique that had been proposed for estimating basilar membrane response input-output function from auditory nerve rate level curves by comparing at best frequency where there's compression on the basilar membrane to off best frequency where it's linear, and looking at the ratio of that. And in fact, you see that the impaired in red, the moderate, the basilar membrane input-output, the estimated basilar membrane input-output was in fact steeper. These were derived from our rate level curves that were shallower. And so, that got us to thinking, and then this is sort of the schematic that we came up with, that it makes sense because, again, there's this inner hair cell between the basilar membrane and the auditory nerve, and with noise exposure, the stereocilia of inner hair cells are just as damaged as the stereocilia of outer hair cells. And so, of course, there's going to be effects there. Less transduction current going through the inner hair cell could lead to, making the things more shallow. And then it's just a confound between inner hair cell and outer hair cell damage. And overall, in our data set, that led to this overall shallower responses. And so, it highlighted the importance of thinking about inner hair cell dysfunction as well as outer hair cell dysfunction. We also looked at, some of the other hypotheses based on, for loudness, how it's encoded in the nerve, looking at summing across the whole population of best frequencies or best frequencies just near the tonal frequency. And this inner hair cell confound really just never let any of those explain the data. And so, the question was, then where does slowness recruitment come from? And after I left the lab, Eric continued with these experiments and looked in the cochlear nucleus and found that for chopper cells, this is sort of the slope of the recruitment slope as a function of the analysis bandwidth that you can use, that the chopper cells had slopes that were consistent with human recruitment slopes, whereas the auditory nerve and primary-like cells in the cochlear nucleus did not. And so it seems like there's some central contributions there. So just to show the legacy of Eric's rigorous and systematic approach to understanding hearing loss and the effects on neural coding, we're alive and well. In our lab, we're looking at just a couple things to highlight here. Inner hair cell dysfunction as another form of hidden hearing loss. Looking at we've used neurometric analyses, again, quantitative analyses relating neural responses to perception, evoked responses to try to disambiguate cochlear synaptopathy from inner hair cells, some spike train analyses we're working on now, trying to estimate the actual inner hair cell transduction from period histograms and then implications for modeling. And then related to some of the effects Ian was talking about with the static vowels, we wanted to think about, is it really just about vowels, or is this a general phenomenon? And of course it is a general phenomenon. And so we've used Wiener kernels to get at temporal coding details and show that to broadband noise and to speech, this upward spread of activity holds as well. So it's a general concept, and we've looked at some non-invasive assays of distorted tonotopy to neural responses to a speech sentence in noise, and compared to audibility and broadened tuning of the tip, this distorted tonotopy, the tails taking over the responses to speech and noise, is the most predictive factor for neural coding. And then in some recent collaborations with Hari Bharadwaj's lab in humans, we're seeing evidence of distorted tonotopy in humans that don't have that much hearing loss, and that it's quite predictive of speech and noise perception. So then finally, just some reflections. First and foremost, Eric was a great teacher. This is a picture well before my time, but of him in a classroom teaching, also in the lab teaching. And I think, you're seeing, the number of independent scientists that have come out of Eric's lab. I think it's really a testament to his ability to balance the hands-on and hands-off training to really make sure that we're developing into independent scientists. And then grant writing, I just can't say enough about how much I learned from Eric about grant writing. Working on his grants, working on grants that I wrote, has obviously been an critically important aspect to me. And then finally, he had just started as editor-in-chief at Jaro while I was there, so there was good and bad about that. It obviously took up some of his time, but that being said, he was at his desk working hard, and you just knock on the door and he just dropped whatever he was doing and would have however long of a conversation you needed to have with him about any issue. But the good part was that he was constantly battling the publisher for fitting papers into limited pages. And so he was very sharp and trying to write efficiently. So as just getting into neurophysiology, the first rate level paper that I gave him, I was nervous, like, am I capturing the points that need to be described for neurophysiology? And sure enough, when it came back, it was totally marked up, every other sentence crossed out in red ink. And I'm like, oh my God, okay, I've got to go back to square one and learn what I'm doing. But when I looked at it, he hadn't changed a single point that I made. All the content was there. It was just making the writing more concise. And he gave, which was a great lesson, and he gave me this lecture on, you can't just ramble around, you have to get to the point, and you just make your points and be done. And I was like, okay, well, some of your early papers were kind of long. Like, for example, the Young and Sachs, the Sachs and Young. And he said, oh yeah, I can barely read those anymore. Those are awful. The science is great, but the writing is just, So it just, made me, one of the ways it made me realize, Eric is just like the rest of us and was training and learning, refining his skills, and gave me, just great lesson in terms of writing. And so, yeah, so it was a great honor to be in his lab and have taken many lessons from him. So thanks very much.
Great. Thanks, Michael and Ian. So our next speaker is Lena Rice from Oregon Health and Sciences University. Lena completed her doctoral work with Eric, overlapping in time with Ian and Michael. And I have to tell you, she was the leader of our small planning group.
I think the next speaker is Ellie.
Okay. Ellie was a member of our planning group, but he was not the leader. So, okay, so Ellie Nelkin from Hebrew University. He was a postdoc with Eric, just coming in as I was leaving. Maybe we overlapped for about a week, and so he completed the canonical DCN circuit by adding the wideband inhibitor characterized by Dr. Palmer over here. And I have to say that circuit was built without a single anatomical experiment. So.
Okay, so. Yeah, I should thank the organizers for inviting me, but it's not. So first of all, I would like to say that I'm really a representative of a large community of researchers who contributed to the story I'm going to say. My tribute to Eric. So Eric, obviously, leader in auditory neuroscience. He was my postdoc advisor for a little bit more than 2 years. And later we collaborated for about 10 more years. And he was always a true friend. And that's, I learned from him more than of any other scientist that I knew during my scientific career. So that's, he really formed me. Now, in 2007, Eric got the Award of Merit of the ARO, and he asked me to present him. And so I collected a few photos that I would like to share with you. So that's Eric I didn't know. You may know that Eric grew up in a ranch, and that he used to say that part of the reason that he went to do science is that it allowed him to avoid waking up at 4 a.m. every morning. Eventually, he found his way to do the auditory research, and this is really Eric, the way that I knew him in the early 1990s at the time that I was in the lab. So my role here is to talk about the dorsal cochlear nucleus. This is, I think about it as Eric's most distinctive contribution to auditory neuroscience. So what is the dorsal cochlear nucleus? What you see here is a dissected auditory brainstem. So this is mouse, this is anterior, this is dorsal. And here is the auditory nerve. We heard now a few presentations about the auditory nerve. The auditory nerve goes into the central nervous system, into this quite large structure called the cochlear nucleus. And you can see from here that it has two parts, okay, the ventral and the dorsal. Okay, when you cut the dorsal cochlear nucleus along this line, then you get something that looks like that. Okay, so you can see that the dorsal cochlear nucleus is a layered structure. There's this molecular layer which is doesn't have many neurons in it. Then there's a dense layer of neurons and the deep layer. And the circuitry is actually quite well worked out, George notwithstanding. Now, there are really two circuits here. One of them is this superficial circuit that looks pretty much like a cerebellum. There are granule cells and these cartwheel cells that are very similar to the Purkinje cells in the cerebellum. I will not talk about it. Patrick will probably say a few words about it later. What I'm going to talk about is the auditory circuit of the DCN that lives mostly in the deep layers. So Eric's achievement was in taking this circuit, actually characterizing the physiological response types of each one of its components, and then figuring out how they connect to each other. And what I'm going to do now is to build these circuits step by step, so that in about 7 to 8 minutes, you will be also, you will understand the DCN as well as I do. So the work started while Eric was in the Goldberg lab, but around 1980, what was known is, first of all, that the DCN is interesting. And part of the reason that the DCN was interesting is that the main unit encountered in the DCN turned out to be dominated by inhibition. So these neurons have high spontaneous rate, and if you use tones of various frequencies and various levels, then it turns out that they are excited by few of these combinations, but they are inhibited by most of the combinations of frequency and sound level. In addition to the type 4 units, there were so-called type 2 units. These are normal early auditory neurons with a V-shaped tuning curve, except for one thing: they dislike broadband noise. Okay, so they don't respond well to broadband noise. So at that time, the circuit of the DCN looked like that. There is a tonotopic axis that supplies excitation to all the neurons in the DCN, to the type 2s and the type 4s, and there is a lot of inhibition on the type 4 neurons. Eric slowly, step by step, figured out what's going on. So first thing was the fact that the type 4 neurons are actually principal cells of the DCN. They send their axons out. Then, together with Herb Voigt, he showed that the type 2 neurons inhibit type 4 neurons. So this is what you see here. This is from the Voigt and Young 1980 paper. So you see here the firing rate of a type 4 neurons locked to the time that spikes in the type 2 neurons occur. These two neurons were recorded simultaneously. And what you can see is a reduction in the firing rate of the type 4 neurons immediately after each spike of the type 2 neurons. So this is direct evidence for the inhibitory connections between them. And together with these type of findings, George Spiro and Eric published this paper that essentially became the canonical model of the DCN, or the first version of the canonical DCN. It had type 2 inhibiting type 4s, and it could account for a lot of the features. For example, there's a bit of excitation at very low sound levels. So it turned out that this excitation at low sound levels had to do because the type 4 thresholds are slightly higher than the threshold of the type 4 neurons. So at very low sound level, these are not active yet, but the type 4 neurons already get their excitatory inputs and they fire a bit. So this is why they're excited here. At higher sound level, the type 2s are excited, and then they inhibit very strongly the type 4 neurons, and you get this central inhibitory area. I think that it took me, as an Israeli, a lot of time to figure out that there was something to understand in this, the use of these initials. Now, this version 1 model had some problems. It did not account for all the effects. For example, why do type 2 neurons respond only weakly to broadband noise? So it turns out the answer was already known. Okay, they had extensive inhibitory sideband. So this is again from the work of George Spiro. What Eric and George did was to play a low-level BF sound in order to raise the firing rate of the neuron, and then they swept another tone as a function of frequency and level, and so you could see now the inhibitory sidebands. So this led to the concept of the wideband inhibitor. Okay, the wideband inhibitor was, at that point, was a neuron that we did not know what its identity is, but we knew what its properties are. Okay? It was weakly active by tones, strongly active by broadband noise, and it inhibited the type 2 neurons. Okay? This is how we conceived of it. And within a couple of years, it was actually identified, first in a couple of papers of Ian Winter and Ellen Palmer, who showed that onset choppers had exactly the right properties in terms of tone and noise responses. And then Donata Ortel and her students showed that these distillate neurons in the cochlear nucleus actually are inhibitory on in the ventral cochlear nucleus, are inhibitory on the dorsal cochlear nucleus neurons. And that accounted for the responses to broadband noise. So with broadband noise, the wideband inhibitor was activated. It inhibited the type 2 neurons. So now the type 2 neurons do not inhibit the type 4 neurons. And what you get is a direct excitation of the type 4 neurons by the tonotopic input. Okay, this is why type 4 neurons respond to broadband noise, are excited by broadband noise. But there was a hard problem, and that hard problem was, again, came from the work of George Spiro with Eric, and that was the inhibition of type 4 neurons by spectral notches. So a spectral notch is a broadband noise, okay, from which we filter out a band of frequencies. Okay? Now, spectral notches are the reason to go there. Spectral notches are important cues for spatial localization. So if we present sounds, for example, from different elevations, you get at the eardrum, okay, you get a spectral shape that has notches whose frequency depend on the elevation of the sound source. So high elevation go with high notch frequency, and the low elevation go with low notch frequency. And what George and Eric found is that the notches actually inhibited the type 4 neurons. Okay, so you see here the response of the type 4 neurons to notches of various widths and different sound levels. And the main thing that you see is a reduction in the firing rate of the type 4 neurons, and they plotted it in these nice Mexican hat-like figures. Okay, each notch got its firing rate plotted at 2 points that corresponded to its shoulders. And you can see here how when you start to widen the notch, okay, the responses actually goes down. And then they used the model, and the model predicted exactly the opposite. Okay? Basically, the model predicted the response to each sound as the sum of the responses to the pure tones that compose it. And then what happens is the broadband noise response is excitatory, and you remove frequencies that are actually inhibitory. So you expect the response to the notch to be larger than the response to a broadband noise. Okay, that's what the model says. Okay, what the data says is exactly the opposite. I want to make this crystal clear. Okay, you have a broadband noise which is excitatory by itself. You take now two stimuli, a notched broadband noise and a narrow noise band. Each of these by itself are inhibitory. Okay, broadband noise is literally the sum of these two. Okay, so you take two stimuli, each of which is inhibitory by itself. You add them together, you would think that you would get even more inhibition, but what you get is excitation. Okay, so that was the problem. And my contribution was basically to show that all you needed to do in order to account for that is to add this additional line to the model. Okay, so that was my contribution. And the way that this works is that with pure tones, the wideband inhibitor is inactive, and so you get the circuit we have been talking about before. With broadband noise, the wideband inhibitor takes out the type 2s, and it inhibits weakly the type 4s, but you still get the excitatory tonotopic input, so the net effect is excitatory. Okay, but with notches, you get rid of this input. Okay, so now let me make it even more dramatic. Okay, so now you get the inhibition that comes from the wideband inhibitor, but you don't get any excitation anymore on the type 4 neurons, and so they get inhibited. So that's the story. As I told you, these type 4, type 2s and the wideband inhibitor have an actual identification in terms of cell types. This circuit is good for identifying cues for spatial localization, but also I think the DCN signals interesting features, generally interesting features of the spectral envelope by this type of sophisticated integration of the information. Now, the DCN is not only this auditory circuit. I want to plot it back in its full beauty. Lina will talk a bit more about sound processing, and Patrick will talk about the multimodal part of the DCN. And so I want to finish by thanking Eric, my mentor and friend, by thanking the wonderful community of auditory neuroscientists who were inspired and guided by Eric in this work, and to you for listening.
Thanks, Ellie. And now Lina Rice. And I just did want to say that we had our planning group, but Lina was really the linchpin who kept us all going and made this happen.
So I guess you gave the intro for me already. Okay. So I'm going to talk today about the beyond the STRF, random spectrum stimuli model. And this was work done not just with Eric Young, but also with other people who couldn't be here today. Shaba Bandyopadhyay, a fellow graduate student, and Jane Yu also, and then Barbara Calhoun and Shans Lee, who were former postdocs in the lab. So in Eric's words, why do we care about input-output function estimation? It is useful to have models that predict the responses of neurons to arbitrary stimuli. So in the periphery, as you saw with Laura Carney's talk, it is possible to develop physiologically based models based on the known behavior and circuitry. But as you go higher up in the auditory system, this becomes more challenging as the circuits become more complex and more nonlinear. So in Eric's words again, it seems more feasible to rely on black box models of signal processing using nonlinear methods derived from Wiener and Volterra analysis. So what are the Wiener kernels or Volterra kernels? Well, the first Wiener kernel is also known as reverse correlation function, and it's essentially the spike-triggered average of the signal. So you can take, say, a white noise stimulus, white noise because it samples all the frequencies that you want to measure, and present it, and record the average, the white noise signal before each action potential or spike. And you get what's called a spike-triggered average. And you get a function that looks like this. In the case of auditory nerve fibers that phase lock, then you can actually get a sinusoidal function. And if you take the Fourier transform and invert that, you get what looks remarkably like an auditory nerve tuning curve. However, it should be noted this only works for a low frequency, best frequency, or BF fibers, as Laura mentioned earlier with the phase locking. So if you have higher best frequency nerve fibers or neurons higher up in the auditory system that do not phase lock, you need the second Wiener kernel, which is called the spectrotemporal receptive field, or STRF. And the STRF generally describes the average stimulus power spectrum as a function of time preceding spikes. You could use a white noise stimulus again. So again, but because these neurons don't phase lock, you have to, instead of averaging the raw signal, you have to average the spectrogram. And so this is a crude STRF crude STRF from the very first paper on this topic using white noise in 1981. And here's an example of the modern STRFs, which you may have seen more recently, using not just white noise, but more rich stimuli, more ecologically relevant stimuli with harmonic components, ripples, maybe constructed in a way to be mutually orthogonal and represent the stimulus space. And then you can construct a more prettier looking STRF, in which case this is the latency and this is the frequency, and this represents excitation at this delay with maybe some suppression or inhibition a little later, and a lower frequency. However, SDRFs are limited in characterization of highly nonlinear auditory neurons. So as mentioned, as you go higher up in the auditory system and things get more complex, you expect more nonlinearity. So one solution is to look at only one domain, the frequency or the time domain alone. And in this case, we focused on frequency. So here comes what we called Eric called these random spectrum stimuli. And there are some parallels to psychophysical estimation procedures of spectral weighting in humans by Green and Berg, or also somatosensory receptive field estimation by Jim DiCarlo back in 1998. And actually, looking back and reviewing this, the first publication in this area related to the RSS is actually Narkin, Kim, and Yang in 1997, which attempted to characterize and predict responses of nonlinear neurons in the DCN. And the innovation in this paper was the addition of second-order weight to try to improve prediction. But this paper was a little early. They were using white noise and pure tones. And then so Eric refined that method to develop random spectrum stimuli shown here. And basically, these are broadband stimuli with bins, stimuli in bins. So they could be like 8 bins per octave. And there would be 8 components per bin to make it more noise-like. And you have these stimuli that vary randomly in their allocation of energy with a standard deviation of 12 dB in the initial studies. And so you could model the discharge rate to any one of these stimuli as a discrete function of that spectral shape. So the firing rate R, now this would be a steady state firing rate for a 300 or 400 millisecond presentation of one of these stimuli, and you record the average firing rate. And then that would be modeled as the sum of a constant rate term, R0, which is the response to a flat spectrum or white noise, plus the weighted spectrum and second-order terms. So the weights W first and W second describe the neuron's first and second-order dependencies on stimulus spectrum. So then you could take the several such RSS stimuli, like say 400 or 800, and then do a linear regression of the weight to all those stimuli by taking all these RSS stimuli, making a matrix S for the first and the second order interaction, and then a vector R and the weight vector and the second order weight vector. And you can use linear algebra and solve for an equation to solve for the weight W and W second. And these weights here together comprise the spectral weighting function of the neuron. So here's some of the early work with Barbara Calhoun, where they looked at auditory nerve weighting function. You can see these auditory nerve first-order weighting functions look very simple. Mostly excitatory, maybe a little bit of suppression, and the second-order terms are mostly confined to near the best frequency. And for Eric, model prediction was very important. So the model is useless for characterizing a neuron if it does not predict the responses. And so here are the predictions for first-order terms here in circles, and this is the rate versus predicted. And you can see that the predictions improve with the second-order terms and shown as the axis, but just slightly. Now moving up the auditory system to the cochlear nucleus, the ventral cochlear nucleus and the dorsal cochlear nucleus. This is work done with Jane Yu, who is a graduate student in the lab. And here are some example rating functions for ventral cochlear nucleus chopper neurons, which are thought to be more faithful representations of auditory nerve firing patterns. And so you can see the rating functions look similar to those seen for the auditory nerve, whereas the dorsal cochlear nucleus rating functions are much more complex. And you saw in the previous talk how complex they are, how nonlinear they are. And these are the second-order terms. And you can see the predictions. Now here, the models were evaluated on prediction of responses to notch sweeps, as you saw in the previous talk, with notches moving from left to right, from low to high frequency. And the prediction to the actual data are the X's and the dotted lines, and the first-order predictions are in the thin, solid lines, and the second-order predictions are in the thick lines. And you can see that the predictions are generally good for the BCN chopper neurons, but they actually bear for the DCN type 4 neurons, even with the second order terms. Compare that with the actual data here. However, the second order terms can still be useful in some cases, and we started ways to characterize, to better interpret these second order terms, these matrices. And so Eric came up with the idea of using eigenvector analysis and to just basically find the eigenvectors with the largest eigenvalues. And you plot them here, just basically multiply the eigenvector by the eigenvalue, and you get a sense of the weighting of the second order terms in frequency space. And you can sort of see here, there's some kind of an edge type sensitivity here. And also in the first order weighting function also shows maybe a sensitivity to a rising edge. And so we did, in follow-up to the earlier work from the previous talk, not only were DCN neurons sensitive notches, but they were also sensitive to rising spectral edges. And so that this second order model helped predict that response. Eric further started to improve predictions using smaller stimulus contrasts and level-dependent functions. So when you have a highly nonlinear function, you can do a piecewise fit. So instead of using 12 dB standard deviation, 12 dB fits over the dynamic range, we could use a 3 dB fit and over more pieces of the dynamic range. And you can see that the predictions improved for the 3 dB compared to the 12 dB. And here are some examples of a level-dependent weighting function, now shown in 3D with frequency and sound level. And I forgot to mention this was work done also with Sharva Bandyopadhyay and myself. And then Sean Lee, another former postdoc, extended that to binaural processing, the computation of both ipsilateral and contralateral spectral weighting functions. So I'm going to wrap this up. So beyond parametric models, Eric also applied information theoretic approaches to studying these nonlinear neurons going up into the inferior colliculus. And a lot of that work was with Steve Chase. And then I'd also like to say that this work was transformative for the field and then encouraged other researchers towards greater rigor in showing that SGRFs and other input-output functions predict the neural responses in order to be considered sufficient characterization and to adopt the use of nonlinear estimation methods in their modeling. And I'd like to wrap up with a few words about Eric Young as mentor and role model. I first started in this lab way back in 1997 as an undergraduate summer intern, 1995 and 1996. And I was so excited to be in that lab as someone with hearing loss. It was so exciting to learn about auditory nerve encoding, speech perception, how hearing loss affects neural encoding. So I ended up coming back to Johns Hopkins for my PhD with Eric Young. And Eric, I just want to make some comments that Eric was a tough yet fair mentor. And I learned a lot working with him between, the introduction to having to present weekly papers, having to present and describe a paper every week and criticize it in the first few years, and then the weekly lab journal clubs. And between all of that, all of us in the lab gained a solid foundation in central auditory neuroscience. And Eric was meticulous. He paid attention to details. And my first paper with him, I thought I was a good writer, but it came back with so many edits. And I can say that that was my first and only paper to be accepted without revision, needing revision. And I don't know if that's common with the rest of you who published with Eric. And in the years after, we kept in touch. I didn't stay in the auditory brainstem field. I ended up going to work in cochlear implants. But we still talk to AOO. And here's a picture of Eric with my then infant son after his Award of Merit lecture and ceremony reception, and my husband. And also, speaking of family, here's Eric with his wife, Pam. And I also remember Pam fondly for, just always being there at the lab dinners and their annual Christmas parties. And I'll just wrap up with what I wrote down in my dissertation, that Eric's high standards set the bar not just for research, but also for the daily interactions of academia. So he's a role model for the right way to be, the right way to interact with people, to mentor people. Thank you.
Thanks, Lena. Our next speaker is Patrick Knoll from Johns Hopkins. Patrick was a doctoral student with Eric and with Paul Mannis. And so I say double the mentors, double the scientists.
Well, thank you. I'm going to shift gears a little bit and talk about the second kind of line of research in Eric's lab, which was non-auditory. It became clear over the years, especially when building these models of the DCN, that the DCN is a very complicated structure, a very interesting and weird structure. And Ellie already described the auditory DCN down here, but you can see this whole part up here. And what became clear over time is that a lot of the inputs to this part of DCN were from other parts of the brain, especially the somatosensory areas. And here's actually a picture of Eric at Aero giving a talk. And yeah. So the story I want to tell is starts in the late 80s. Anatomists started to wonder what the inputs to the DCN were, and they did very simple experiments. So this is a slide from one of these papers from Ito. They put retrograde tracers into the DCN and looked where the cells that projected DCN came from. And they found that there's a bunch of cells right here in the somatosensory brainstem, like in the cuneate nucleus and the spinal trigeminal nucleus, that send basically monosynaptic inputs to DCN. The question is, what are these inputs doing? Eric, of course, jumped on this right away, being in the DCN, having developed a DCN model version, I guess 1.0 at the time, trying to figure out what in the world is going on. Why is the DCN getting somatosensory input? So the first set of experiments was very simple by today's standard, very difficult then, was basically to poke around and stimulate the areas that got input. So Eric and Ellie and Ruth took cats, recorded in DCN type 4 neurons that have very high spontaneous rates, and then they poked stimulus electrodes into the somatosensory nucleus to stimulate the area where these cell bodies have been found. And they found if you stimulate this area, for example here, you see a very strong inhibition of the DCN neurons by somatosensory, by this input. You need very little stimulation. Oh man, my eyes go bad. I think it's 40 micrograms something. So it's very, very sensitive input. They also found that if you stimulate while you present auditory information, auditory stimuli, you can inhibit the auditory evoked response. So there's a very strong inhibitory input coming to most of these neurons, but some neurons were also excited. The question then was, where is the input coming from? So they stimulated multiple different areas in the cuneate and the gracilis nucleus and found that inputs that could be activated by the pinna. So when you touch the cat, you could also record from the stimulus electrode, and they found spots that were responsive to the pinna gave the strongest response, suggesting it may be something to do with the ear projected to DCN. So of course, this was model 1, right? So we want to see, Eric wanted to see how the DCN has processed information. So he developed a computational model of the superficial DCN that could replicate some of these currents that they recorded. So they did LFP recordings to kind of figure out how this signal processed, and they came up with this computational model, which was of course a computational model, a compartmental model, based on like a raw model. And they figured out that there has to be a neuron up here, like the cartwheel cell, that inhibited the pyramidal cell, and that both the cartwheel cell and the pyramidal cell would be activated by these parallel fibers in the superficial layer. And their model replicated the LFP recordings perfectly. Of course, it was Eric. But there was a model, so you had to somehow validate the model with physiology. So 2 postdocs in the lab, Roger Miller and Kevin Davis, started to follow up on these experiments to record in the superficial DCN. And they did a modified stimulation paradigm where they basically stimulate the MSN, so the dorsal column nuclei, 4 pulses. I'm trying to see if there's an irritation of the response. If they can infer from the irritation of the response which component of the response reflected activity of some of these circuit components. And they found that in the first component, you get a very strong inhibition Second stimulation, you get inhibition with a little bit of excitation now of the DCN neurons, and so on. And the reason that they found that basically this first strong inhibition and this excitation always covariate when they're stimulated. So the reason that they had to share the same input, reflective of this direct parallel fiber input to the fusiform cell and to the cartwheel cell. And they saw the same components when they stimulate the parallel fibers directly, suggesting it's this pathway that transmits the somatosensory input. This still kind of left then the cartwheel cell as the central element of the circuit. Then somebody had to go in and record the cartwheel cell. This was very difficult. These cells are on the surface of the DCN. They have very small cell bodies. They're pretty hard to record from. So Kevin Davis figured out how to etch electrodes. At this time, we etched our own electrodes, and you could shape the tip of the electrode to record from particular neurons. So Kevin figured out how to etch electrodes to actually record from cartwheel cells into the recording from one of these cells. So when Kevin then stimulated the dorsal column nucleus and recorded, he found that cartwheel cells are indeed excited. So now, if excitation to cartwheel cells, these cells are inhibitory. So these are the ones really inhibiting the type 4 neurons or pyramidal cells. And here you see superposition of one of these cartwheel cell responses with a response to type 4. You can see cartwheel cell excited very strongly at the first pulse. Type 4 neuron inhibited very strongly. At this pulse, you can see second pulse, cartwheel cell has adapted, and you can see now this breakthrough excitation, which is basically direct input of the parallel fibers onto the DCN type 4 neuron. So that was great. Now we had kind of completed the top of the circuit. The question was, what is this input doing, and where's it coming from, right? And this is where I entered the fray. You can see me here as a young graduate student at the rig, the same place that you saw Eric with a cat. I was an engineer. I came from chip design. I made microchips, but was interested in biology. And I was attracted to Eric's lab, having lots and lots of technology and toys to play with. So we would sit here for many, many hours at night. These were the old days with a cat, like you record for 24 hours. And as a young graduate student, Eric would sit right there, and we would record together. And as you will see in 2 slides, that was kind of important for some of these experiments to have 2 people in the room. As Ian already mentioned, there was a seniority in the lab in terms of what desk you get. This was the desk Roger Miller had before as a postdoc. When he moved on, I got to move to his desk. And being a techie, I liked to have computers and lots of toys, and Eric and I connected very strongly on that level. And I ended up having 2 monitors on my Mac then, which was in the 90s, right? You didn't have 2 monitors normally, but I had 2 monitors and I could display data on both of them, which was great. I graduated in 2000, went off to do a postdoc, but I returned back to Hopkins 20 years later. And this is one of our faculty picnics during COVID. There's me, there's Eric, there's Larry Schramm, who ran the graduate program when I was a student. I'm now running the graduate program, so it's kind of a circle of life, which is beautiful. During COVID, Eric still came to the lab every once in a while, and we would have lunch, and we went to the Eastern Market to have fried chicken, which was very healthy, of course. And this was our last outing, December of 23. Eric came to the lab, we had chicken, we spent a couple hours talking science, and then he went home. So where are we going with this? So I was an engineer, and at Hopkins in the BME program, we take anatomy. And so it's human anatomy, you dissect a human. So I got very good in dissecting, and we had gone through dissecting out peripheral nerves. I was like, hey, there's this somatosensory input. Can we trace it out? So I decided to trace out the peripheral nerves innervating the DCN that give rise to this input. So these are very long dissections because you had a cat and you dissect all the peripheral nerves, record the DCN, and then stimulate all those nerves. Eric did not allow cauterizers in the lab because he grew up on a ranch and he was reminded of branding cattle. So you had to be a very good surgeon to not have a lot of blood there, right? Because then that would be bad. So I had to tie off a lot of blood vessels. And so I started stimulating all these nerves in the periphery. And what I found is that cervical nerve number 2, the one innervating this side of the head, gave the strongest input to the DCN. So if you stimulate cervical nerve number 2, you record DCN, you get very large evoked potentials. If you record from type 4 neurons, it looks exactly the same as it looked when you stimulated the dorsal column nucleus. So that was kind of a big clue of where this input is coming from. You could also stimulate the trigeminal nerve, innervating more the lower part of the jaw and the tongue and those kind of things, but it also gave a very strong input. So then what was the input? So it's cervical number 2, especially the branch innervating the ear, but what's the input? And this is where the two people come in. That's why it's so important where Eric was in the lab. Because what we started to do is to manipulate the ear and trying to figure out what is the input. And it turns out if you just push down on the ear of the cat, so here's the cat, the famous neuron coding lab cat by Phyllis Taylor. If you just push down on the ear like this and kind of stretch that tendon that the cat has there, moving the ear, then you got a very nice inhibition of the DCN neuron sitting right here. The problem is you have to be inside the chamber at the cat, but also you have to record being outside at the PDP-11. So you need 2 people. And the experiment literally done counting to 10, right? So you're in here, 1, 2, 3, 4, 5, push, 1, 2, 3, and so on, right? Of course, this was then not sufficiently quantitative for Eric. He wanted a way to make it more quantitative, so I built a little stimulator, like a cat ear stretcher, which is basically a loudspeaker with a stick and a cable attached to the ear, that then I could sit outside and program that. So when you stretch the ear in a repetitive fashion, you can see a beautiful inhibition of the type 4 neuron right here. Okay, or since I also learned how then to etch these electrodes like Kevin, I could record from cartwheel cells, you can see cartwheel cells. So this kind of fit the picture, so it has something to do with muscle stretching. So it has to be a muscle receptor that projected into the dorsal nerves. And why would you want to have that? Well, Ellie already told you that maybe the DCN decodes spectral notches. The notch depends on the relative location of the sound to your ear. So to make sense of the notch, you have to know where the ear is. So maybe those 2 streams of information are associated. Okay? So that brings us back to the kind of second study that I performed in Eric's lab, which is how are these 2 inputs working together. So I followed up on the initial work that Eric did, presenting sound and somatosensory stimulus at the same time, but just roving the stimuli around. And what I found is that if you stimulate the dorsal column nuclei a couple of milliseconds, 10 or 20 milliseconds before a sound stimulus, you can boost the response of sound. And other times you could suppress the response of sound to really like it has a long latency or long-term kind of very nonlinear effect there. I also want to show you the nonlinear effect to publication. The early papers that Eric published came out very soon. This is 2011. I graduated in 2000. I was a tenured professor by the time it came out. And what had happened is that, we had collect all the data and analyzed, and then it was just sitting there. Eric got busy, he had moved to other things, and he wanted to reanalyze the data. And that, of course, he's very meticulous. So he kept reanalyzing the data and got busy with other things and other things and other things. But at some point, we published this. I'm still very proud of this paper. So Eric moved on to the IC. For him, the story was done. But others picked up his data. For example, here is a paper by Susan Shore, who basically replicate everything we've done in the guinea pig. So it's not just cats, also guinea pigs have the somatosensory input. And the question is this useful at all, right? Could this somatosensory input contribute to something? And there had been floating around at the time some ideas that maybe the somatosensory input has something to do with tinnitus. And Robert Levine, who was at the Mass Eye and Ear Institute, had seen some patients that had somatosensory evoked tinnitus, where you could, push on the neck and stuff, and the tinnitus would change. So he always suggested there might be something there in the DCN. And at the time also there was a hypothesis that as hyperactivity in the DCN contributing to tinnitus. So Susan Shore then jumped on this and followed this up and tried to see if you can pair maybe somatosensory stimulation with the sound stimulation. Can you suppress some of the DCN activity? And maybe that can help with tinnitus. So here's the initial paper where they did some pairing. You can see at certain latencies, you can see some suppression, other latencies, some enhancement of DCN activity. But out of this came a company, and they have a device that looks like this. It doesn't look very impressive, it's just a stimulator, but you have to stimulate at just the right time. But you can see the tinnitus score that this treatment actually helps. So it can suppress tinnitus somewhat. Of course, there's a second company doing something similar to the tongue. And remember, the tongue is innervated by the trigeminal nerve. So it might be the same circuit actually being underlying the kind of treatment effect of both of these devices. So with this, Eric didn't think about treatment, right? Eric was just driven by trying to understand what the heck the DCN is doing, what the circuit is, right? So it's like really basic curiosity-driven science that others picked up to really then build companies and actually help patients, which I find amazing. You just go off, right, to where's the circuit coming from, right? It's like a true scientist. Down here, you have Eric with Murray Sachs. At my graduation, you were at a Hopkins party. Eric and Pam, and yeah, you saw that picture already, part of that. With that, yeah, Eric was a great mentor. I mean, we spent a lot of hours recording from cats together. I feel very guilty because I don't have that time to spend with my students. I cannot spend nights with my students recording from an animal. We also don't do 24-hour experiments anymore. And then the telephone call would always come from Pam, like it's time to come home, and he had to pack up and leave. And then it was just me at the night and the Baltimore police, choppers flying around. But it was great times, and it's, I think, highly unusual for somebody who was so busy as he was. He was, doing the journal stuff, everything, to kind of sit there for many, many hours and really hanging out. And you get to know somebody very well. When you sit there, through a night with an animal, you record, you talk about all kinds of stuff, and you build a great relationship. So he became a great mentor throughout my career, and I think it's a great honor that we're colleagues too. Thank you.
Thanks, Patrick. Our next our next speaker is Alan Palmer. Alan is emeritus professor, which is why he looks so relaxed. But when he was an active professor, he was the director of the MRC Center for Hearing Research in Nottingham in England, which at the time was worldwide one of the key centers for auditory system research. He had quite a powerful team. His work overlapped, was complementary to Eric's, as you'll hear. They had a long relationship as colleagues. And we're glad, Alan, that you were able to extract yourself from the comfort of retirement and join us today.
Thank you very much. Good evening. My name is Alan Palmer, and as you see from my affiliation, I retired some years ago from the University of Nottingham. Prior to that, for about 45 years, I was an auditory neuroscientist. And most of that time was spent at the Institute of Hearing Research in Nottingham. And during that time, sure enough, Eric Young's research interests and my own overlapped on many occasions. And I think that's why the organisers, and I'm still not quite sure why, felt that I was an appropriate person to come here today and talk about Eric's international influence and indeed his influence in ARO. So let's jump straight into it. He says, trying to work this. The most important factor in determining a scientist's international impact is their work published in high-quality journals, peer-reviewed journals. There's no question about that. And what's more, it's the only metric we've got. So, how did Eric do? Eric Young's publications have attracted nearly 13,000 citations. I've only rounded that up a little bit, giving him an H-index of 59, which most of you know, and those who don't, means 59 of his papers got at least 59 citations. And his even now, his papers are attracting 260 citations a year. Now, this is a flawed metric. Everybody knows it's a flawed metric because it depends on all kinds of things. One of which is the size of the congregation that's going to cite your papers. If you're in imaging, you get thousands of citations. Eric worked in a niche area. These numbers are really, really good. And for any auditory neuroscientists working in this area, these are good numbers. They're about as good as they get. So, Eric had a major international impact. But whatever way you look at it, this is playing the long game, the really long game. Now, in this digital age, you publish a paper, you advertise it on X and Meta, and someone on the other side of the world downloads a PDF and reads it. Even then, it's a long game. You think of the idea, you apply for a grant, you do the work, you analyze the data, you write the paper, you submit it for publication, goes through peer review, and eventually that person downloads the PDF and reads it. So even in this day and age, there's a long time. And after he's read the paper, to cite the paper, he's got to write a paper. So you're talking about a lead time now, probably of, 2 to 5 years. Back then, everything was on paper. Everything went via mail. So the lead time to actually get a citation was, it could be 10 years. Now that's ridiculous. Science cannot progress via this particular route of someone reading your paper. There has to be a quicker way. And the quicker way is what we're doing here. You go to international meetings, you go to scientific meetings, and you present real-time transmission of information. Now, that's all very well, and it was very easy for American scientists back then because Society of Neuroscience, ARO, Acoustical Society, great meetings, okay? So you could stay, Americans could stay in America, get their point across, make their impacts, and all the rest of it. And for me at least, all of those meetings, including ARO, were essentially American meetings to which foreign scientists were very welcome. And I cite the fact that I was on the International Committee of ARO. No international society needs an international committee to look after their international members. I don't know whether they've still got it, but you know, whatever way you look at it, essentially these are American meetings for Americans, and we're allowed to come to them, and we're very grateful to do so. So, is there another way to get at the international audience? Well, you go to international meetings. Come on, come on. So, did Eric Young actually attend and contribute to international scientific meetings? Well, of course he did. Here's one in 1991 in Salamanca. On all of these, just look for the little circle, and that's Eric. Very characteristic, usually at the back, very tall. You can pick him out. This one's Spain, 1991. This one, I believe, is probably San Francisco, but I'm having a bit of trouble. So that was also an international meeting. Come on. Okay. This one's back in Spain again. This one's in Bad Nauheim in Germany. It's on the my right-hand side, your right-hand side probably. That one's Utrecht in the Netherlands. That one's somewhere near Bordeaux in the south of France. That one's the Royal Society in London. That one's Grantham in England. That one's Prague. And that one's, I've no idea. And that one's in Kloppenburg in the Netherlands. And that one's in Ascona in Switzerland. It almost looks like I'm stalking Eric Young, doesn't it? But I'm not. This is not actually a catalogue of Eric's international conferences. I've no idea which ones he went to. This is a random selection. And the way it was selected is these are the photographs that were pinned to my notice board when I retired a long time ago. And the reason they were on my notice board is the eagle-eyed amongst you will recognize the fact that I'm on every single one of these. These are the ones that I went to, and Eric was also there. What you also notice is that these are quite small meetings. These are everybody at the meeting. There were less than 200 people. But they were genuinely international meetings. They were held in different countries all across Europe, but they were never dominated by people from that host country. They were people from a lot of Americans that you will probably recognize on these pictures, although they're a little small. They were from Australia, New Zealand. They were from all over. So they were genuinely international meetings. And Eric actually attended these meetings and talked to people, gave the talks at the meetings. Where am I going with this? These were quite small meetings, as you can see here. This is actually a couple of pictures. This one's one in Prague Castle. And if you look carefully, you'll see Eric and Murray in the front row. And about 4 rows behind that, there's me talking to Kent Marest. The point about these small meetings is they're only one session. It's not like these parallel sessions here. Everybody attends every single paper, and you're there for days or a week to actually accommodate that. Now, Eric actually contributed to these meetings much more than I did, much more than I did. And the reason for that is what I like to do if I had a question, I'd kind of track down the speaker afterwards in the bar or whatever and ask them face to face what I wanted to know. Probably it was a lack of confidence, believe it or not. But Eric asked questions. He asked a lot of questions. And everybody knows people who get up and ask questions at every paper. It's because they, sometimes want to hear the sound of their own voice. Sometimes they want to score points. Sometimes they want to say, I published that 9 years ago or whatever. That wasn't what Eric Young was doing. What Eric was doing, he sat there through every single paper, and this was psychophysics, it was physiology, it was clinical, it was the whole gamut of hearing science in these meetings. And he appeared to just hoover it up, actually understand everything anybody was saying about any subject. And at the end of it, he'd get up and ask a question. And those questions were apposite, they were penetrating. They were never nasty, he never belittled anyone. So when Eric got up to ask a question, I listened, and everybody else in the room seemed to listen as well. So he enriched these meetings enormously by asking questions. And because he was asking questions pretty indiscriminately, whatever the subject was, he had an influence well outside of his niche area of dorsal cochlear nucleus or whatever. Now, there was one other way that Eric influenced things. When I was a PhD student, my supervisor came back from a meeting in Edinburgh in Scotland. And he was really excited because he'd spent a lot of time talking to Eric Young. And that was the first time I think I'd ever heard Eric Young's name, to be quite honest. Now, what's surprising about that? Well, you think nothing, but actually, this is 1975 we're talking about. Now, Eric Young, at the time, was a young man, new into the area. I don't know how long he'd been in the business, but not that long. My PhD supervisor was a man called Ted Evans. Now, Ted back then was actually the most respected auditory neuroscientist in the UK and possibly Europe. He was really at the top of his game just then. So he's gone off to a conference, and he's come back all excited because he's talked to some young guy from the States, and he's at the top of his game. So Eric was having influence on the international community right from almost the start of his career. And he says, trying to get this to work. Here's another example. Here we all are, sitting in the middle of a street in Salamanca. We went to Salamanca quite a lot. Salamanca's a wonderful place. So who you see here? Well, there's me, a lot more hair, a lot less weight. There's Ian Winter, ditto. There's Mike Hewitt, there's Ray Medis, Enrico Mugnani, Eric in the middle, Joe Adams, and unfortunately, I'm old, I can't quite remember the other two guys' names. But the point is, this is what we did. You were stuck at these meetings, and I don't mean stuck really, because they were in nice places. You were at these meetings, and they'd finish, and then you'd have to go and find dinner. You'd have to go and find a bar, because we often stopped for a beer on the way. You broke up into small groups, and you sat around talking informally, and you'd talk about anything, but you'd talk about a lot of science. And for me, at least, these were almost the most enjoyable parts of the whole meeting. Talking about science informally. And Eric did a lot of this. So, come on. There were 4 ways at least that Eric Young actually had an influence internationally. First was definitely the publications. But as I say, you've got to be around a long time. To have an H factor of 59, you've got to have published 59 papers, and you don't do that in a year, unless you're Brian Moore. Come on, there we go. He presented his work at these meetings, so he's presenting the information, getting it across in real time. He asked erudite and apposite questions that illuminated whatever had just been said for me and the rest of the people in the audience. He really enriched these meetings. And finally, he did an awful lot of informal networking with anyone and everyone. So you think that would be enough. But actually, Eric had one last ace up his sleeve. If these international scientists didn't actually come round to his point of view, he had one last thing he could try. He could send him one of his boys to make them an offer they couldn't refuse. I think you all recognize Georgie the Muscles Spirou and the incalcitrant neuroscientist Ray Medis. Okay, so let me give you an example of how Eric directly influenced the work of my lab. This is the paper where George and Eric said, we got this thing, we're calling a wideband inhibitor that's got these properties. but nobody's ever found these in the cochlear nucleus. Fine. But at the time, I was working with Ian Winter. Now, Ian Winter was my first PhD student. He went off to Australia, came back, and he was working with me again as a postdoc. And we were looking at the properties of a group, large stellate cells in the ventral cochlear nucleus, for reasons I won't go into. But they seemed to have the properties these guys needed. They described the cells we'd actually characterized. So I went to Johns Hopkins, and I said, look, here's our data. This is what they do. They're just what you're looking for. Maybe they're a candidate for the wideband inhibitor. And they said, yeah, thank you very much. Then there was a paper by Bill Rohde. And Bill said, yeah, people have suggested that these large stellate cells are actually the wideband inhibitor. They appear to go to the dorsal, but nobody knows where they go. So I thought, well, we ought to be able to do something about this at least. So what I did was I sent another of my postdocs, Robert Arnott, off to Didier Pinot's lab to learn juxtacellular labeling. He came back and taught the rest of us how to do it. And we applied that technique to the large stellate cells to try and find out where they went. Okay? And this is actually, I'm going to try and stop this, but I may not. The green bits are the cell body and the dendrites, and the purple bits are the axons. And as it rotates, if I fail to stop it, those of you in the audience that recognize the cochlear nucleus will see the projection that I was looking for. Oh, no, it's done exactly what I didn't want it to do. Okay, try again. Come on. No. Yeah, I tried all this morning and it worked fine. Oh, no. It seems to have jammed up. I seem to have broken it. Can you restart it or something?
Maybe somebody can help out.
Yeah,
Nothing's happening, so.
Yeah, don't worry. Sometimes it does this here. Which one were you? The last one?
The last one, that's
Okay,
it.
right there.
Okay, and I just whizzed through them, shall I?
Yeah, pardon.
Okay.
Sorry about that.
It's all right. Thank you.
Don't go away. Don't go away.
I'm getting the wrong way, am I? Yeah. Just refreshing you.
There we go.
Now, can we-
Can you tap in the middle? Move the cursor to the middle.
Well,
There.
okay.
Yeah.
I'll just rotate that.
Just let it go.
Go. Oh, it's rubbish, this, isn't it? All right, I'll just let it go round. I can tell you in words anyway. We demonstrated unequivocally with this technique that the cells in the ventral cochlear nucleus projected to exactly where we needed them to be in the fusiform cell layer of the dorsal cochlear nucleus. I could have told you that in words anyway, but the reason I was showing this graphic is because I spent hours and hours and hours looking down a microscope to do this reconstruction, and this is the last time it'll ever get shown. Okay. Oh, now I can't even get rid of it, sorry. I'm doing well here. You can tell I'm a bit out of practice. No? Try this. That's done it. Okay, so basically we'd shown that we have cells with the right characteristics and that they actually went to the right place. That's about as far as we could go. It was still circumstantial information, evidence, but it was good evidence, and we'd gone about as far as we could with the techniques in the lab. But the technique of juxtacellular labeling actually proved to be pretty useful. So we went on from there to study the lamina structure in the inferior colliculus, and as this goes round, you'll see it all goes into a single lamina. and the columnar structure in the auditory cortex. So you could, at a stretch, say that Eric actually influenced all these other projects that took place in my lab, because I wouldn't have had that technique in the lab at all if I hadn't been trying to show that the wideband inhibitors were the cells we were looking at. Here's another example. This is taken from Dan Presnitz's paper. This is a small network that was put together for them to explain responses in the ventral cochlear nucleus to co-modulation masking release signals. What you see, just looking at it, is they've got the wideband inhibitor right in the middle of it, okay? Clearly a pinch from the dorsal network, an obvious influence. However, I should point out this is a cheat. This is not an independent observation. Dan and Raoul are genuine card-carrying French citizens who work in Paris. But Ian Winter is also an author on this, so it's not quite an independent observation, this one. So, when I agreed to give this talk, I sent round an email. I said to my friends and colleagues, I said, look, I'm giving this talk, I've got no idea what I'm going to be saying. You got anything I can use? So I got some replies. And here's one of them, from a guy called, David McAlpine. And Dave said this: people who know how important his approach to the field was to you, meaning me, and how that shaped us through your mentoring. It's worth recognizing in that context. Without being morbid or trite, I told people for many years that the ghost of Eric Young sat on Alan's shoulder. Your insistence with me, no doubt an Eric thing too, on obtaining every possible classical measurement of a neuron before investigating the experimental paradigm in question, made our 2001 paper on frequency-dependent ITD processing something we weren't looking for, and my career from that. Notice the last line. Now, there's two things to this. One is, I thought that was a bit strong. But then when I actually thought about it, in one way it was absolutely correct, which is that whenever anybody working in the peripheral auditory system on structure and function, you wrote the paper, and if you wanted to send it off to a high-impact journal, there was a really high probability the manuscript would end up on Eric Young's desk for review. So it kind of kept you on your toes. It was like he was looking over your shoulder, and you crossed the T's and you dotted the I's. Now, to be fair, we do that with any paper anyway, but that was an additional stimulus. And the other thing is that last line. This kind of suggests that Eric should take some of the credit for launching David McAlpine's career. All I can say is, Eric, if you listen, you can have the credit, but you've got to take some of the responsibility for it too. So, Eric Young's impact of the ARO. We've already heard most of this, and I'm going to do it again, if I can get this. Eric was a staunch supporter of ARO. He attended the annual meeting every year, and along with his students and postdocs. They gave posters and whatever. He did exactly at ARO what he did at the international meetings. He gave talks, he asked questions, and he networked. That alone would mean he had a major contribution to ARO. But he was also a good citizen. He served on the awards committee. ARO thought a lot of him. 2007, they gave him the Award of Merit. He came back again to chair the ARO Award of Merit Committee. And in between, as you all know, he was the editor-in-chief, the founding editor of Jarrow. Now, that's an interesting one. Jarrow formed a publications committee to find out whether it was viable to have a journal. When they decided it was, they put it out to a vote of the members, whether or not we should start a journal from the association. I voted strongly against it. You may ask why. There were good reasons. Up to that point, specialist hearing journals struggled a bit for content. They had contracts with publishers, which meant they had to publish stuff, and as a result, they published some really good stuff and sometimes some pretty poor stuff. So you wondered whether there was enough good stuff out there for another journal. And then there was Auditory Neuroscience, which was created to try and cure that problem and didn't last very long for different reasons. But that demonstrated to me, A, there might not be the stuff out there, and secondly, that you can't guarantee that a journal is going to be successful. Now, the other factor was that previous to this, ARO had moved its midwinter meeting from Daytona. And that caused severe financial problems for the organization. It could even have been, endangering the existence of the ARO, because it was quite serious, I believe. And that worried me. I was concerned that a really successful journal would be good, a source of income, but one that wasn't successful might be a financial burden. Largely reflects the fact that I don't really understand publishing contracts. But anyway, that's why I voted against. And then you, the members, in your wisdom, voted 2-1 for the journal, so it was created. And then the Publications Committee did this. Finally, the Publications Committee considered a potential first editor for JARO to recommend to the Executive Council. They agreed that the person selected be well known to the membership of ARO, be a highly esteemed scholar with interests that span those of the ARO membership, and have a strong reputation for leadership and collaboration. The idea of recommending Eric Young rose quickly in the discussions, and he was a unanimous choice as the first ARO editor-in-chief. Now, Eric wasn't the only person who could have done this job. This organization has loads of really talented scientists who have managerial and editorial experience. But I think it's very, very noteworthy that when the Society wanted someone who was going to be a figurehead, that was going to be absolutely reliable, absolutely respected, and set the tone for this new journal, that they turned to Eric. Being Eric Young, he had absolutely no difficulty in recruiting his founding associate editors. Look at this list. There's some really big names down there, and oh, you look, I'm on there as well. The minute they decided, we decided we would have a journal, and they appointed Eric, I was all for it. I was going to do whatever I could to help it be successful. Eric phoned me, I came running. I was on there for about 5 years. The tenure's usually 3, but because everybody was appointed at the same time, Eric didn't want to sack them all at the same time. He wanted to stagger it slightly. So I was on there for about 5 years. By the time I took this job, I'd reviewed hundreds of papers for dozens of journals and grants and all this kind of stuff. So I wasn't exactly green, but I really enjoyed and learned a lot working with Eric in those 4 or 5 years. And one of the reasons I enjoyed it so much was because of the ethos of the new journal. And that was put forward by Eric in an editorial in the first edition, which said, yes, we're going to reject papers to keep the quality up, because you need a high-quality journal, but also the editor and the associate editors are going to work with authors to try and make sure their papers are good enough to go in this journal. And that was an approach that I really, really identified with and approved of. So, my job here today was to tell you about Eric's international influence and his influence within ARO. And I hope, I think, I've really demonstrated that he had a big impact in both areas. I'm going to finish on a rather more personal note, I hope. My first professional visit to the USA, I flew into Boston. I was a day late. I gave a seminar there a day late at the Eaton Peabody Laboratory. I flew down to Baltimore. I gave a seminar at Johns Hopkins, and I went on to an Acoustical Society meeting in Austin, Texas. You have no, I was young. I was in my early 30s, starting out in the career. You have no idea how nervous I was about that trip, meeting Nelson Kiang. His reputation preceded him. Nelson was really nice to me, and he always has been every time I've met him. But coming down to Baltimore to meet Murray Sachs was the one I was really worried about, but also Eric Young. God, it was really... However, I arrived in Baltimore, and they were lovely. It was a terrific visit. They made me welcome, they were friendly, I gave a seminar, they were interested in the science, they asked nice questions, probing questions, but not nasty questions. They sent me around the lab to talk to the postdocs. It was a great visit, the sort that you really want. And subsequently, I came to ARO every year for about 30 years, and it was nearly always in Florida. And whenever I had interesting science to impart, I'd stop off in Baltimore and give them a seminar and talk to them. So I'm really pleased and honoured to say that in all the times I met Eric at conferences, at ARO and in Baltimore, that he was much more than a colleague. He was a real good friend of mine. I'm pleased to be able to say that. I'm going to leave you with a final quote. This quote is from my very first postdoc, a man called Adrian Rees. I think many of you know Adrian. He's gone on to bigger and better things, as most of my postdocs seem to manage, but that wasn't difficult leaving my lab. Anyway, he sent me this quote. I'm not going to actually read, well, I could read it to you, but I was going to just leave you to read it yourself, because in my opinion, this succinctly tells you what members of the international community, and probably the domestic community, thought of our friend and colleague, Eric Young. So thank you very much for listening.
Thank you very much, Alan. And then our last speaker is Tilak Ratanatha, who's going to talk about the Eric Young Memorial Fund. And Tilak's been very involved in the educational mission at Hopkins, worked with Eric, and Eric was one of his faculty mentors.
Thank you. I would like to talk about the Memorial Fund, and I'm very pleased to tell you that we have raised $27,000. And thank you to many of you who have given money. And I'm going to tell you what Lena and I and a few other people are going to do with the funds. In addition to being a mentor of many of them, Eric was the first advisor for the first student with hearing loss to do a PhD in BNE, Lena, Auditory neuroscience. He was a positive influence and encouraged other PIs to recruit and mentor students with hearing loss. Now, 25 years later, there are about more than 150 current and past trainees with hearing loss in the Hearing Inclusive ARO group. Many have progressed to other areas of STEM. ARO includes now 14, I added one more today, including this first student and plus 2 up and under coming PI with Kangaroo early career grant who will contribute to the period. And now, what the goal of the fund is, in the United States and in some countries, trainees with hearing loss in STEM obtain support via supplemental grant, while before this, the current administration and trainee grant, likewise before this year, fellowship or scholarship for foundation or accessibility grant. Here, the ultimate goal is to provide support for any trainee with hearing loss in STEM, first in the US and later worldwide, that otherwise could not be provided or rectified by the usual mechanism. I'll give you a couple of examples, but they're not exhaustive, but I'll give you a couple of examples, facing some of the problems. Registration and travel costs of conferences. One-time accessibility costs for lab seminar, like a Chromebook or desktop, for Bluetooth microphone, for automatic speech recognition app, and diaper for research experience to post-back medical students not covered by current funding DEI grant that we have now, and accommodated for interviews and depth exam, medical career exam. Here, there's an important example, cost of minor but urgent repair of hearing aid or cochlear implant or replacing battery not covered by insurance. And a final example I would like to give, the cost for hearing aid. There was a medical doctor who came to the US for a PhD. Hearing loss was notified the advisor who bought hearing aid for that student and completed residency and fellowship in neuropath and now faculty in the medical school. So thank you for all the donations, and please give us some idea how we should. We have a lot of other new ideas about spending the money in the next few years. Welcome. Thank you.
All right. Thank you all for coming to the symposium. And a round of applause for all the speakers.